Okay, this is a chapter on antibiotics and chemosynthetic agents, and it's not too difficult, I don't think, so you shouldn't have much of a problem with it. The chapter is called Antimicrobial Drugs, um, so let's get into it. All right. Um, I'm going to talk about these people in class, so I'm not going to bore you on this tape with these people. These are key people in the development of antimicrobials. You can see very easily that the development of antimicrobial drugs, we're talking about antibiotics and synthetic agents for treatment of infectious diseases, is a 20th century discovery. Nothing before that, really, other than the first thing you see, which is quinine. Quinine uh, has been around for hundreds of years, used by the Indians of South America for treating malaria. But obviously they didn't know what the cause was. They didn't know how it worked. It was almost as if magic, almost herbal, really, than anything else. Anyway, the rest of these drugs are all in the 20th century. I do have a couple of pictures, though, over on the right. I don't think we may or may not have already done this lab before we get to this lecture, but you're going to be seeing these pictures in lab, actually based on the plates that you make in lab. So up at the top you've got a mold growing. This happens to be penicillium mold in the middle of the agar plate. And you've got staph colonies that have been streaked in the background. However, right around the area of the mold, you see no colonies. Notice where I'm drawing the blue lines right here. There seems to be an area in which no staph colonies grow, or hardly any staph colonies grow. So, obviously, if you saw this in the 1900s and you were scientists, it would make you think that there must be something being made by this mold right here that is blocking the growth of the bacteria. Otherwise, they wouldn't be avoiding the area around the mold. Obviously, that was penicillin. The mold is called penicillium. And of course, down at the bottom is the application of that understanding, which we will be doing in lab, where we streak a microorganism, put antibiotic discs on it, and we look for that zone, the area around the antibiotic disc where the bacteria are not growing. And that will tell us whether the organism is sensitive or resistant to the drug. But there's one other thing that's kind of interesting here. We're also going to be talking about drug resistance later on. And notice that there's some tiny, tiny little colonies in that clear zone around the penicillin disc. Those are drug-resistant bacterial colonies. We'll talk more about that coming up. Wanted you to notice that there are a heck of a lot of microorganisms that make antibiotics. The key guys you see right here. But there are many more bacteria that make antibiotics. Many of them are used all the time. Like Pseudomonas makes an antibiotic called mupirocin. Cannot remember its brand name, though. That's its generic name. And as well as other bacteria. But the four big boys of antibiotic production are 2 moles and 2 bacteria. The 2 bacteria are Streptomyces and Bacillus, various species of Streptomyces and various species of Bacillus. And for the fungi, there's Penicillium mold and Cephalosporium mold, its cousin, very similar to Penicillium. But of course, the big ones are Streptomyces, Penicillium, and Cephalosporium. Bacillus is not a big player in antibiotic production, although I would venture that most of you have got these antibiotics in your bathroom. Many people buy triple antibiotic ointment to use on wounds and such, And that's usually bacitracin, polymyxin, neomycin combinations, and bacitracin and polymyxin are made by Bacillus. The pictures that you see around here, over on the left up at the top is Streptomyces, and down at the bottom right is the mold Penicillium. Before we go on, wanted you to notice how many different antibiotics Streptomyces makes. And that's just a short list. I could have added more. Okay, so first let's talk about the groups of drugs. An antibiotic, let me define it, is a chemical made by a microorganism that inhibits or kills other microorganisms. Think about it. Think about why they make this. If you're a bacterium and you live in water or you live in soil, and there are millions of other cells growing around you, if you have the ability to make a chemical that will kill them, you're now at an advantage. You get more nutrients, you get more oxygen, you get more space. Life is good. So, antibiotic production evolved in bacteria to inhibit competitors around them. Now, there's another group, the opposing group, that's made in a laboratory from scratch. These are totally man-made. They are called synthetic or chemosynthetic drugs. We'll talk about examples of each category. Then we've got a group called the semisynthetic, which are actually based on true antibiotics like penicillin, but they have been manipulated in a laboratory by rearranging the molecular structure to become a different antibiotic. For example, penicillin can be manipulated chemically and turned into ampicillin, carbonicillin, piperacillin. There are a whole variety of different kinds of penicillin derivatives. These are made in a laboratory, but they're based on the natural penicillin formula. Now, I'm going to be using generic names. I wanted you to understand the difference between them, though, generic versus brand. Over on the left, you see generic names like Pen G, Penicillin G, Ampicillin, Cefalothin. Those are generic. Over on the right are trade names, brand names, and they are developed by a pharmaceutical company. So you could have 3 pharmaceutical companies marketing gentamicin, and one might call its brand garamicin, another one might call it Genticin, another one might call it Chemical G, whatever. But it's all the same generic drug. Okay, mechanisms. Basically, these mechanisms, there are less than 6 of them. So on this overview, let me point out over here the guys that hit cell wall synthesis. We're going to talk about those. We're going to talk about the drugs that do damage to the cell membrane. We're going to talk about drugs that are called metabolic analogs, that is, they block a metabolic cycle. That's these guys right here, the sulfa drugs. Then we've got the guys that in some way block or retard protein synthesis, those guys. And then finally we've got the guys that do some damage to DNA replication, and that's the guys at the top. So as you can see, there are about 5 major mechanisms right there. Okay, so let's talk about cell wall synthesis. This is the penicillin family. Everything in the penicillin family works exactly the same. Remember, peptidoglycan. It's in the walls of almost all bacteria. It's in gram-positive walls, it's in gram-negative walls, but there's more of it in gram-positive walls because gram-positives have more Pg. The way that these drugs work is they block cross-linking between the amino acid chains sticking off of a peptidoglycan structure. Remember that the PG structure is a very kind of a lattice-like structure. I likened it to wood pallets. If you know the wood pallets in the grocery store that they bring boxes in on. If you stacked about 6 of those wood pallets together on top of each other, you would have a kind of a similar structure to peptidoglycan. Now, extending down, linking the pallets together, or linking the PG layers together, there would be like twisty ties or chains of amino acids holding all the pallets together. And that's where penicillins and cephalosporins, because they work the same, work. They block that cross-linking, so it makes for a very weak PG, and in fact it creates holes in the PG. So as the cell is trying to grow to get bigger, to divide into daughter cells, these small little holes, they start out as small holes, but then the holes get larger and larger as the cell is trying to increase in size, and it can't fill these holes with PG because of the damage done, and eventually these holes become large holes and the cell disrupts. Okay, then we've got a big group of the protein inhibitors. And these work in a variety of different ways depending on where they work in protein synthesis, because remember, you're starting with mRNA, messenger RNA. It has to attach to the ribosome. It has to fit into an active site in the ribosome. The ribosome recognizes a codon on the mRNA. A tRNA comes in with amino acid. It has to attach to the mRNA via its anticodon. The ribosome has to move down one spot and start the recognition of a new mRNA codon. Remember also there's a start codon. Remember there's a stop codon where the ribosome falls off the mRNA. So finishing up a polypeptide chain. So there are a lot of places where a drug can stop protein synthesis. So these drugs, I'm just going to say they block protein synthesis, but really there's some very subtle differences in where they block protein synthesis. Now, you remember that protein synthesis in bacteria is pretty much like protein synthesis in your cells, with a few variations. One of the major ones is remember bacteria have different ribosome structures. However, because protein synthesis is pretty much protein synthesis, many of these protein synthesis inhibitors will also affect our cells, because our cells do pretty much the same thing. So there are some side effects to many of this group. But you see 2 of the big guys, streptomycin and all of its family, and tetracycline and all of its family. All right, the metabolic pathway inhibitors, the sulfa drugs. Sulfa drugs, things like Bactrim, SXT, Gantracin. You've probably taken one of these. They're very often given for UTIs, for urinary tract infections. Okay, so in order to understand this, you need to know something about folic acid. All right, so let me point out right here, folic acid. Folic acid is needed by cells to make DNA and RNA. Your cells need it. Bacterial cells need it. Your cells get it in your diet or in supplements. A lot of people take pills. But you get it in your food. Bacteria make their own from scratch. But in order to do that, they need a chemical called paraminobenzoic acid, PABA. You see PABA right there. It's not important to know the structure. Now, remember, eukaryotic cells, like your cells, get it in their environment, get it in their food. They don't make it. It's preformed. That's going to be important. So, notice that there are some molecules over here. These are 3 examples of sulfa drug synthetic agents. By the way, these are all synthetic. They are not natural. Let's take sulfanilamide. Look at sulfanilamide and look at PABA. In fact, look at any of them and you'll notice that they have a core structure that is similar. So, if you look over here at this big blue guy down here at the bottom, here is an enzyme. And this enzyme is using PABA to make folic acid in order for the cell to make DNA and RNA. If there is one of the sulfa drugs present, it will be picked up instead of PABA. The enzyme, of course, is blocked. This is a competitive inhibitor. So in other words, when sulfanilamide is attached to the enzyme, PABA cannot be attached to the enzyme. Therefore, you get no folic acid. No folic acid, no RNA, DNA. Cell dies. That's the way these guys kill bacteria. They don't affect your cells because your cells don't make folic acid. They get it preformed. Very good group of drugs. Okay, the guys that do something to DNA. There are a lot of variations with this. Let's go with the quinolones first. This is a big group. You've probably taken some of these before. An example would be Cipro. That's a brand name, by the way. Ciprofloxacin is its name, real name, generic name. You might have also taken Levaquin. Levofloxacin is its generic name. You might have taken moxifloxacin, or moxifloxacin is its generic name. So this is a big group. Have a lot of toxicities, but very effective at killing bacteria. The way they work is in bacteria. Remember, bacteria have one circular double-stranded DNA. It's supercoiled. In order for the cell to replicate itself, the DNA has to be uncoiled, unsupercoiled, and then we have to nick off one of the, open up an area of the circular chromosome, and it unrolls, and the cell makes two daughter strands from the original strands of DNA. That uncoiling of the supercoiled DNA requires an enzyme called gyrase, G-Y-R-A-S-E, and the quinolones block that enzyme. So the DNA can't be uncoiled, it can't be replicated, the cell can't replicate. All right. Now, the other guys you see up here, the nucleotide analogs, like acyclovir, like Famvir, Gantvir, I'm trying to think of other variations of it. The only two I can think of right now. Valcyclovir is another one. If you look over here at all the variations over in this area, these are nucleotide analogs. They look like DNA nucleotides. Remember your adenine, guanine, cytosine? That's what we're talking about. So in the presence, if these drugs are present, and by the way, these are all antivirals, and remember, a virus gets into a host cell, and the host cell actually reads the DNA or the RNA, some viruses are RNA, and replicates the nucleic acid using nucleotides made by the host cell. Host cell's doing everything. Well, if you throw some of these drugs at the host cell that's being taken over by a virus, the host cell will pick up some of these chemical formulas instead of the correct nucleotide, like a guanine or a cytosine nucleotide. And obviously, the nucleic acid of the virus is not going to be what it should be, because we have chemicals being substituted for the nucleotides. And that's how it blocks viral replication. All right, then we have some odd and end other things we'll talk about a little bit later in more detail. Cell membrane permeability. Cell can't control what goes in, what goes out of the cell. Polymyxin, that's one of those drugs that's in a triple antibiotic ointment. Then, when we get into flu, we'll talk about this group more, but prevention of viral attachments. Viruses cannot be treated with antibiotics. There are no targets for those antibiotics. I've taken you through things like cell membranes and protein synthesis and ribosomes and DNA uncoiling and cell wall, and viruses have none of those targets. So any of the antibiotics that I've just mentioned, useless. So we're talking about a whole different group of drugs used for viruses. If you can even treat a viral infection, many of them cannot be, like rabies. So over here on the right you see a host cell being infected by, in this case, flu virus. And it's kind of interesting to note how the flu virus goes through its takeover of a host cell, because if you understand that, you can see how the drugs work. So over here is the attachment of the virus to the host cell. And there are some drugs that can be given that will block that attachment. If you can block the virus from attaching to the host cell, it can't get in. Then, another thing that the virus has to do is it's got to insert its nucleic acid into the host cell so that the host cell can replicate the nucleic acid and read the genes to make viral proteins. There are drugs that block that, like AZT, you see over here on the right, does that. Then, the nucleic acid has to be replicated. It has to be read. The genes have to be read to make viral proteins. These are viral proteins being made right there. And then those viral proteins are going to be assembled into a whole virus, which then can escape the host cell. Look at all the antivirals that can block that. This is a big group right over here. These are called the protease inhibitors. Again, we'll talk about them more in viruses, but that's the big group that's given for HIV these days. So again, the bottom line is viruses require a very different group of drugs. So, if you're in the market of making drugs, you're a pharmaceutical company, these are things that you need to think about. You want a large toxic dosage over therapeutic dosage ratio, which means, let me give you an example. Let's say that the dosage for drug X that causes toxicity is 500. That is toxicity to the host. Might make you have diarrhea, might destroy your bone marrow, might do liver damage, might cause deafness, whatever the toxicity. That's the toxic dosage right there. Now this is a ratio, right? I'm going to put it in a fraction. Therapeutic dosage, let's say, is 250. This would be the dosage that would get rid of the infection, all right? So that ratio is 2, correct? 500 divided by 250. Now that's drug A. Let's go to drug B. Drug B, let's say, has a toxic dosage of 300, and its therapeutic dosage is 275. Let's say this is milligrams. Well, now your ratio is only about like 1.1, whereas up here it was 2. So which one is better? The first one that I gave you with a ratio of 2. If you look at the numbers, you'll notice that the spread between 250 and 500 is bigger than the spread between 275 and 300. In other words, you want as big a gap between therapeutic dosage to get rid of the infection and toxic dosage that will cause problems in the host as possible. And if those two dosages are the same, like let's say it was 1, that would be a totally useless drug, because then you'd be causing damage to the host while you're getting rid of the infection. The toxicity could be worse than the infection itself. So you do not want to do that. Okay, other things you have to think about. Readily available, inexpensive, chemically stable, easily administered. You've got to be able to get it where you need to get it in various parts of the body. You'd like it to be selectively toxic. You don't want your drug to kill off all the bacteria in the person's body, because most of the bacteria in your body are good. They're making proteins for you. They're making vitamins in your gut. They help turn on your immune system to pathogens. They do more good than negative. If you decimate all of your normal flora, you're going to have problems in a week, 2 weeks, 3 weeks that again could be worse than the actual infection you're trying to get rid of. You'd like your drug to be non-toxic. You'd like to be non-allergenic. Toxicities. Not going to talk about this much. I'll talk more about it in class. So you might want to stop this and take a look at it. Other things. You need to know the spectrum of action. I'll give you an example. Well, I think I can do it maybe now. So on this chart at the top, you see spectrum of activity. Notice that some drugs, like penicillin, mainly used against gram-positive. That's their biggest group right there. Whereas the sulfonylamides, or the sulfa drugs, work across the gram-negatives into the gram-positives, into some of the other kind of weird bacteria like the chlamydia and the rickettsia, and sometimes even into protozoa. If you look at streptomycin, very good against gram-negatives, as well as Mycobacterium. This is the guy that causes tuberculosis, and it's gram-positive. Look at this drug over here, niclosamide, used very, very sparingly just for worms, and only some worms, like tapeworm. That's narrow spectrum. So we have narrow spectrum drugs, we have broad spectrum drugs. Efficacy. This is more of what the pharmacist needs to think about, but you need to think about dosages, how you're going to give it. Different drugs may work better IV, may be good oral. Some drugs are destroyed orally, like Pen-G cannot be taken as a pill because it's sensitive to stomach acid. You've just wasted some money taking it, and it doesn't get rid of your infection. Side effects, drug interactions. Some drugs work together well. Some drugs cancel each other out. And remember that the patient may have other problems that they're taking drugs for, like diabetes or cardiovascular problems. So now you have an antibiotic or synthetic agent interacting with one of those drugs, or you may have an antibiotic A interacting with antibiotic B, where you get less effect because you're giving two drugs together that counteract each other. Okay, so how do you evaluate a drug? When a new drug is developed, whoops, just went by itself. When a new drug is developed, how do they evaluate for efficiency and whether it works? Or you go into a hospital, you go into a clinic, you have an infection, that sample, specimen, has to be taken, has to be sent down to the microbiology lab. They have to run an identification set of tests on it. They have to run an antibiotic sensitivity test on it to determine its sensitivity. The doctor may have already started prescribing a drug, but they still have to do an antibiotic sensitivity. Okay, different ways to do it. Kirby-Bauer, we're going to be doing in lab. This is a Kirby-Bauer test that you see on the picture. Bacterium has been streaked in the background on the plate. Antibiotic discs are placed on the plate. They're incubated. You can see the organism growing in the background. You can see that some of these drugs seem to have very large zones, like this guy, this guy, this guy, that seem to be inhibiting or killing the bacteria. So the organism appears to be sensitive to 6, 8, 4, maybe 1, but certainly not 7, certainly not 2, not 3, not 5. Obviously, you wouldn't want your doctor to give you drug number 2 for this infection, whatever this organism is, because drug 2 wouldn't do anything. That's the easy test. However, that's not the most common test that's done. The most common test is called the MIC, Minimal Inhibitory Concentration Test. where dilutions of an antibiotic are done. Here are dilutions of tetracycline. So you see over here, tetracycline is our drug. And notice the concentration. So start over here. High concentration, 50 micrograms. Then this would be 25. You can't see it, but that's 25. Here's 12.5. Here's 6 and 6.3, 3.1. Notice that they've diluted this 1 to 2, 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 because you can see the dosage is being reduced as you go down. Then the organism that is causing the infection is added, and we're looking to see if the organism can grow. So notice that the organism is growing, 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 not. Okay, the MIC, minimal inhibitory concentration. The minimal dosage, the minimal concentration of the drug that will kill or inhibit, and that would be this guy right here. You get any more dilute than that, going to the left, the organism starts growing. Okay, another useful test is called the beta-lactamase test. Now, to understand this, you need to understand what a beta-lactam is. Look at the structure of penicillin. It's right here. There it is. Here's its cousin, cephalosporin, on the right. Notice that there is a green part of a molecule right there. It's called the beta-lactam ring. No matter what kind of penicillin drug you're giving, all penicillins and their derivatives have that beta-lactam ring. Now, the side chain might be different, like ampicillin versus carbonicillin, but they both have that beta-lactam ring. And so do the cephalosporins. So everybody, all the drugs in these two families have the same basic structure. Well, penicillin and all of the family, again, have these beta-lactam rings. And this beta-lactam ring is susceptible to being destroyed by an enzyme that some bacteria can make called penicillinase, or what are sometimes called beta-lactamases, because they go by different names. But penicillinase is the one that is against penicillin. It's an enzyme made by the bacteria. Again, the bacteria has evolved to become resistant to a drug. If the organism, and notice I put down here staph species because they are some of the most egregious at making these enzymes. If the infection is being caused by staph, and if that staph has the production of penicillinase, and if the doctor gives penicillin for the treatment of this infection, it will do nothing. The infection will go on, person could die. It will not get any better. So obviously the doctor needs to know this pretty quickly before you give the wrong drug. Penicillin and its family are some of the most often given drugs along with the cephalosporins because there's very few side effects and they're very inexpensive drugs. But if an organism makes beta-lactamases, you don't really want to give this group of drugs. So there is a very easy test to do called the beta-lactamase test, and I'll take you through that in lecture. So it identifies that enzyme so that the doctor can change his or her mind about a drug being given, or change a drug. All right, there are other variations of evaluation tests for drug sensitivity. These are this is one that's gained some momentum in the last few years. It's called an E-strip. It's kind of a combination of an MIC and a Kirby-Bauer dysdiffusion test. These numbers that you see, this happens to be probably tetracycline, that you're looking at. You see a big T right there under the E. And then you've got some numbers here. These are concentrations of the drug. So highest level, most concentrated up here, less concentrated here, less, least. So you've got decreasing concentration of the same drug, one drug. So notice you got an organism strict in the background. You have a zone of inhibition right there where the drug is the most concentrated. But then as the drug goes down, 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 the organism starts growing. So what you do is you read that dosage right there where there's no more kill or inhibition. Over on the right, you see the same thing, except they've run 6 E-strips with 6 different antibiotics. Side effects, allergies, destruction of normal flora, all problems related to the drugs that we're talking about. Again, many of these problems are worse than the actual infection itself. Sometimes not, but sometimes yes. If you look at some of these toxicities, like go down here to hepatic injury, liver damage, that's really severe. And there are a lot of people on liver transplant lists, so it's not likely that you're going to get one. Some of them not too bad, like diarrhea. You could live with diarrhea for a few days. Renal injury, kidney. I've already mentioned damage to your own normal flora. Allergies we're going to talk more about when we get into the immune system. Allergies are a peculiarity of the person's immune system. Okay, antibiotic resistance. I'm going to stop right here.